Very good evening to all the TG EAPCET aspirants whoever have appeared in the previous exam, and now we are going to deal with TG EAPCET 2025 exam paper that has happened on 29th shift 1 paper. Okay, so this session is going to be very much useful to all the EAPCET aspirants of Andhra Pradesh, and most of the concepts here we are going to brush up with very good memory hacks and mnemonics.
Yes. So let's start our session with botany first, the next zoology, and we'll be discussing chemistry at the end of this session.
Yes.
Okay? So let's continue with our session. And students, so we like to request you that, see, there are very few students are watching, so
yes,
you can also share with your friends, those who are seriously preparing for EAPCET,
yes,
especially from Andhra Pradesh.
Yes.
Okay? So this will be giving us some boost to do more videos and more informative content for you. So take this as a consideration. So let's start our session.
Yes. Whichever you feel like, you can drop in the comment section. You can also travel with us for this complete session. So if you look into the botany first question, okay. So here this question is from cell biology. Okay, this question is from cell biology wherein different stages of cell cycle are given and we have to correlate them and match it. First is synapsis. Okay, synapsis happens in zygotine stage, exactly correct, which leads to bivalent formation wherein homologous chromosomes come together forming a synaptonemal complex or tetrad formation, bivalent formation that happens during synapsis, that happens during zygotine stage, and crossing over occurs during packaging, which leads to recombination of the genes by the enzyme recombinase. So yes, 1 is true and 2 is also correct. When it comes to replication doesn't happen in diplotin, replication occurs in the S phase, okay, S phase. So this is wrong, okay. So third one is wrong. Fourth is terminalization, that is diakinesis. In diakinesis, terminalization of chiasmata happens, but here there is no division of chromosomes, So if you can see, if you can look into the correct match, that is option 1 and 2. 3 and 4 are wrong because replication is not occurring in the diplotin, it is of S phase, and terminalization of chiasmata takes place in diakinesis. Terminalization of synaptonemal complex occurs in diplotin. So yes, here we have to look for the correct answer, that is option 3. And then if we look into the next question, this is from morphology of flowering plants, where we need to consider both assertion and reason. So in assertion, if you consider, Acheranthus will show spike inflorescence, true, Acheranthus will show spike inflorescence. Spike shows basipetal arrangement of sessile flowers, wrong. Spike doesn't show basipetal arrangement, spike shows acropetal arrangement of flowers. Acropetal arrangement of flowers, not basipetal arrangement, because spike comes under what inflorescence beta? Racemose inflorescence. So here the answer is option 3, assertion is true but reason is false. And next if we consider, this is again from morphology of flowering plants, that is stem modification, subaerial stem modification. Runners is one of the subaerial stem modification, wherein the stem of the strawberry and subaerial stem of the oxalis are the examples for runner, okay, not underground stem, that is subaerial stem of strawberry and subaerial stem of oxalis are the best examples for runner, wherein chrysanthemum and jasmine, these are the examples for sucker. Okay, these are the examples for sucker. So here if you look into, the right option is option 2. Okay, and next if we see, unilocular, that is one chambered ovary, becomes bilocular, that is two chambered, due to the formation of false septum is seen in. The formation of false septum is called as replum. False septum is nothing but replum, which is observed in the family Brassicaceae. This is observed in the family Brassicaceae. Okay, so the examples for this family Brassicaceae is given, that is mustard and argemone. That is what? Mustard and argemone. And next, if you consider anatomical characters applicable to hydrophytes and xerophytes respectively are, okay, anatomical characters for hydrophytes and xerophytes respectively are. In hydrophytes, if you consider, they are poorly developed xylem. Hydrophytes, you will see poorly developed xylem, and in xerophytes, if you can observe multi-layered epidermis with cuticle coating over it. So here this is option 1 will be your correct answer. And next question, if you consider which of the following is prevented by unisexual flowers that present on different plants? Okay, unisexual flowers, you can say some synonyms for this beta. Unisexual flowers are also called as dioecious. Unisexual flowers are also called as dioecious or heterothalic. Okay, these are the synonyms for unisexual. Best examples for unisexual, consider like papaya, date palm. These are the examples for unisexual flowers. In unisexual flowers, when male reproductive structure and female reproductive organ are on two different plants, definitely only xenogamy occurs there. Only xenogamy, that is cross pollination. Okay, only xenogamy occurs, that is cross pollination. Autogamy and geitonogamy are not occurs. This doesn't occur in unisexual plants, because autogamy, that is pollination between same flower, geitonogamy, pollination between two different flowers of the same plant. This will occur only in the bisexual or monoecious flowers, not in the unisexual flowers. Fine. Next question, if you consider beta. Consider the following statements like assertion in Liliaceae flowers are homochlamydous. What does the word mean or the term mean? Homochlamydous. Homochlamydous means that both calyx and corolla are indistinct. They are indistinct. Okay, both calyx and corolla are indistinct to perianth. So in Liliaceae, flowers are homochlamydous. Heterochlamydous means they have two different calyx and corolla. In Liliaceae, perianth is differentiated true. In Liliaceae, perianth is differentiated wrong. In Liliaceae, perianth is not differentiated. Okay, perianth is not differentiated in Liliaceae. So here if you look into reason is false but assertion is true, then we have to go with the option 3. Okay, reason is false and assertion is true, we have to go with option 3. Next, if you look into, this question is from biomolecules. Okay, this question is from biomolecules, wherein identify the below structural formula along with its related function. Okay, in one complete page of our Telugu Academy, this is given, this structure is given, all the structures of biomolecules, be it nitrogen bases, different carbohydrates, proteins, they are given, amino acids. Okay, so one such structure that we have observed is of lecithin. Okay, this is of lecithin. Lecithin is a major component of phospholipid in the cell membrane. Okay, lecithin is a major component of phospholipid in the cell membrane, that is option 3. Okay, lecithin is the major component of phospholipid in cell membrane. And next, if you consider we have to select the correct pair from this. Genetic nature of DNA is not given by Frankel and Conrad, it is given by Watson and Crick. And in vitro DNA synthesis, it is not done by Hanning, and double helical model of DNA is given by Watson and Crick, not RNA. Lastly, if you see genetic code is given by Marshall Nirenberg. Okay, genetic code is given by Marshall Nirenberg. Fine. So this is option 4. Genetic code is given by Marshall Nirenberg. Next, if you see identify the character of ovules to the bean family. Bean family means leguminous family, that is Fabaceae. Okay, for Fabaceae. Okay, in Fabaceae, if you see, first to identify the characters of ovule, you should know what type of ovule you will see in the Fabaceae family. In Fabaceae family, you will see Campylotropous ovule. Okay, Campylotropous ovule. You can see Campylotropous ovule, wherein body of the ovule will bend. Body of the ovule will bend, and ovule is placed at the right angle to the funicle. Ovule is placed at the right angle to the funicle, which is the stalk of the ovary. Next, embryo sac is slightly curved to horseshoe shaped. Micropyle comes towards the funicle. Yes, micropyle comes towards the funicle. So here we have to choose option 4 when all the 4 statements are true. Next, if we see, again a tabulated form from morphology of flowering plants. You can see how many questions are asked from that chapter, which is very important. First is Pisum under Fabaceae family, which has diadelphous stamens. True, they have diadelphous stamens, but it is not 2 carpellary or bicarpellary. It does not have 2 carpels, it just has 1 carpel. Okay, it just has 1 carpel. So you will call it as monocarpellary. You will call it as monocarpellary. Fine. And next, if you see, that is Arachis. Next, if you see Arachis, just a second. Next, if you can consider, that is Arachis, wherein monodelphous stamens are given. No, this is not monodelphous stamens, that is diadelphous stamens, because this is also from the same family. This has diadelphous stamens. And dehiscent pod, yes, this is the type of fruit that you will observe, beta. This is the type of fruit. Next, Nicotiana, panicle inflorescence and fruit capsule. This is true. Next, Ruscus, unisexual flower, and the modification Cladophil. These 2 are true, but Arachis is wrong with respect to stamens. Pisum is wrong with respect to carpel. So in the question, you have to identify clearly that they have asked us the wrong pair. When wrong pair is asked, we have to choose 1 and 2, that is in option 2. Okay. Next question, if we consider, very important, focus here. Okay, wherein cell division and this is a connecting dot between 2 chapters, that is cell division and fungi. Okay, cell division and fungi. So in this you have to know one thing. Agaricus basidiocarp is having 60 basidia. Okay, it is having 60 basidia. Find out the number of nuclear fusions, meiotic divisions, and basidiospores formed from that basidiocarp. Okay. So here, if you can see in Basidiomycetes, basidia is the site of meiosis. Okay, if you see Ascomycetes and Basidiomycetes, Ascomycetes and Basidiomycetes. Ascomycetes and Basidiomycetes, site of meiosis in Ascomycetes is ascus. In Basidiomycetes, it is basidium. Both of them are diploid, beta. Both of them are diploid. Do remember that, because this is the site of meiosis and these are the meiocytes. These are what? meiocytes. So here inside the ascus or basidium, what happens? Nuclear fusion happens and meiotic divisions happen, forming the basidiospores. Okay, if 60 basidia are present, 60 nuclear divisions and 60 meiotic divisions will happen. 60 nuclear fusions and 60 meiotic divisions will happen. And how many basidiospores will be formed? 60 into 4. Okay, those are 240. Okay, 60 into 4, those are 240 basidiospores are formed. Basidiospores, or these are meiospores. Fine. So here, if the same thing is asked with respect to ascus or asci, same process you have to follow. So here if you see 60, 60 and 240, this will be the right answer. And here what you have to remember, what are the meiocytes? Okay, here the meiocyte is ascus and this is basidium. And next, if you consider, yeah, this is from Cell, the Unit of Life, wherein identify the correct statement regards the Golgi bodies. So if you can see, Nana, from nuclear membrane, that is nuclear envelope, is elongating as reticular structures. These are elongating as reticular structures. These are endoplasmic reticulum. Towards the endoplasmic reticulum, the convex face of Golgi, is present like this. Okay, convex face of Golgi. So the convex face or cis face, or forming face is towards the endoplasmic reticulum, and towards this side it is transphase or mature phase or concave phase is towards the cytoplasm. This is the mature phase. So here if you can see, convex side is cis or forming phase, true. Concave side is called trans or maturing phase, that is correct. And cisternae of Golgi can modify the proteins, yes, into glycoproteins, and it modifies lipids into glycolipids. Materials enter through the trans phase and released through the cis phase. Wrong. Materials enter through the cis phase and they release out through trans phase. So this is wrong, Nana. All the three statements are true. The fourth statement is incorrect. So here what you have to choose, 1, 2 and 3 are the correct statements, which is option 3. And next question is from plant kingdom. Okay. So here if you see, identify the true statements given below. True statements. Okay. 1. Gymnosperms have albuminous cells and sieve cells. True. And phloem fibers are made up of collenchyma cells. Wrong. Phloem fibers are made up of sclerenchyma cells. Okay, phloem fibers are made up of sclerenchyma cells. Next, phloem parenchyma is absent in most of the dicots. Wrong. Phloem parenchyma is absent in most of the monocots. Most gymnosperms lack vessels in their xylem. Wrong. Yes, most gymnosperms will lack vessels in their xylem. So here what are the true statements are 4 and 1. Okay, these two are wrong because this is in the monocots. So here you have to choose option 3. Next question, ma. Life cycle of rockweed. Stay down here and look into the question properly. Life cycle of rockweed. Okay, rockweed is the common name of fucus. Okay, of fucus. So if you look into the most common life cycle of algae is haplontic life cycle. Haplontic life cycle is there for algae. But there are few organisms which have exception for the haplontic life cycle of algae. For example, fucus. Fucus has diplontic life cycle. Fucus has diplontic life cycle. Okay. And if you consider few more examples like ectocarpus, kelps and Polysiphonia and Polysiphonia. Okay, Ectocarpus, Kelps and Polysiphonia, they have haplodiplontic life cycle. They have haplodiplontic life cycle. So here if you see, the diplontic life cycle is seen in fucus. Common name of this is rockweed. So we have to choose option 2. Next question. Here if you see the simplest amino acid, okay, simplest amino acid is glycine. Simplest amino acid is glycine. 16 carbon fatty acid is palmitic acid. Same from biomolecules the question is asked. And simple lipid is trihydroxypropane. Aromatic amino acid is tryptophan. Tryptophan is the aromatic amino acid. So here the option will be 2. That question is from biomolecules. And if you see what are the quick referral systems in the taxonomic studies, when these 4 options are given, you have to choose herbarium because it is the quick referral system. In the absence of this herbarium, you can go with key. Now if we see, match the following type of question again, list 1 and list 2, wherein cycles shows what type, okay, cycles shows first thing coralloid roots, okay, cycles will show coralloid roots, that is association with blue-green algae, that is cyanobacteria, Next, Anthocerus capsule has pseudo-elaters, and Markantia capsule has pores and elaters. Salvinia is heterosporous one. Along with Salvinia, any of the heterosporous forms beta, that are Selaginella. Okay, Selaginella, Marsilia and Azolla. These are few others along with the Salvinia, which has heterosporous condition, which is first observed in the pteridophytes. So this is option 1. And next question if you look into, this is with respect to peroxisomes. Peroxisomes are the small microbodies which are single membrane bound that you will observe in the inside the cell, both plant cell and animal cell. So these peroxisomes are involved in catabolism of long chain fatty acids, role in photorespiration through, and hydrogen peroxide destruction. All this are related with peroxisome. Which is not related is synthesis of glycolipids. Synthesis of glycolipids and glycoproteins, they are related to Golgi bodies, not peroxisomes. And next, match the following type of question, wherein Golgi apparatus will help in the formation of glycoproteins and glycolipids. Golgi bodies will help in the formation of glycoproteins and glycolipids. Lysosomes help in the digestion of lipids. Next, vacuole helps in osmoregulation. Glyoxysomes, conversion of stored lipid to carbohydrates. So here if you look into the options beta, that is A4, B3, C2 and D1. Next question, this is from genetic code. Simple only, just by looking into the known genetic codes are only asked most of the times. Now also it is asked in that way only. So if you see AUG, that is the start codon and it is coding for methionine, which is option 4. Okay, AUG is coding for methionine. UAA, UAG, UGA, they are coding for stop codons, that is end codon. Okay, and UUU is coding for phenylalanine, and UGG is for tryptophan. These 3 are most noted for us, and based on that we can easily identify the last one. This is very important, wherein if you see, it is as clear as it is. Just by looking into the question you can answer better. In pea plant, Mendel studied 7 pairs of contrasting characters. The ratio of pairs of contrasting characters that are related to flower, pod and seed. Okay, with respect to flower, he has considered 2 traits. What are they? Flower position and flower color. Second, pod shape and pod color. Seed shape and seed color. So these are the two traits. So if we take the ratio of that, it is 2:2:2. So this is the ratio of this. Next, if you see, here, this is from strategies wherein we have to identify the plant, its varieties. So here they are asking for the mismatched pair. Just by looking into that, you can answer that for flat bean it is not Pusa A4 and fruit borer. It is not that insect pest on that. So next it is option 4, that is flat bean Pusa Gaurav and the insect pest is aphids. So these two are mismatches. This we can directly answer looking into the diagram or the table from the strategies. So these two are mismatches that are found here. And next, this if we see the final acceptor of electrons in the light reaction. In the light reaction, final acceptor of electrons, that is from Z scheme, non-cyclic photophosphorylation. Final acceptor of electron is NADP+. NADP+ is the ultimate electron acceptor. This by the enzyme NADP reductase. By the enzyme NADP reductase, it forms into NADPH plus H plus. Okay. So here this is option 4. And next if you see, this is most important question. I think most of the students felt difficulty when you are answering this, but it is very simple. This is just based on 3 points. If we are following, then we can answer this actually. So yes, the length of DNA is 510 angstroms. He said it has 20% of 6 aminopurines. We have to find out the number of nucleotides and number of hydrogen bonds in that DNA. So here if we see the normal length of the DNA, if we take for one turn, okay, for one turn, one complete turn, the length of the DNA is length of one turn, okay, one turn, or we can say one pitch, or the pitch of the DNA is 34 angstroms. For one pitch, it has 10 base pairs. It has how many base pairs? 10 base pairs. And here we have given 510 angstroms, how many base pairs are present? 510 angstroms, how many base pairs are present? For that it is very simple, just 510 by 34 if you have to take, then you will get to know how many turns is there in, how many turns will be formed in 510 angstroms length of DNA. How many turns? 15 turns. It is 15 turns. 510 by 34 is 15 turns. If it is one turn is having 10 base pairs, 15 turns will have 150 base pairs. 15 turns will have 150 base pairs. And we know one base pair is having 2 nucleotides, right? One base pair is having 2 nucleotides. 150 base pairs will have how many nucleotides? 150 into 2, we have 300 nucleotides. Out of 300 nucleotides, 20% of it is 6-aminopurine, that is 20% of them are adenines, he is saying. Okay. So 300 into 20 by 100 will give you 20% of 300 nucleotides. That is nothing but it is 60 nucleotides. When 60 nucleotides are adenine, 60 nucleotides will be thymine, because adenine pairs with thymine by 2 hydrogen bonds. 60 plus 60, it is 120 nucleotides. 300 minus 120 is 180. The rest remaining nucleotides are 180 nucleotides. Of those nucleotides, we have 90 nucleotides of cytosine which binds to guanine by 3 hydrogen bonds. Again here 90 nucleotides. This is how you have to fill the complete set of nucleotides. It is very simple, ma. If we are having 3 points in our mind, one turn is 34 angstroms and each turn is having 10 base pairs, that is the second point. Third is one base pair is having 2 nucleotides. Just looking into these 3 basic points we can apply, and that is how we are getting the answer. So now we got how many nucleotides are present, we got it. But adenine pairs with thymine by 2 hydrogen bonds, that is 60 into 2, 120 hydrogen bonds here. Next, guanine pairs with cytosine by 3 hydrogen bonds, 90 into 3, 270. So on total you will get 390 hydrogen bonds. The total number of nucleotides are 300 and the total number of hydrogen bonds is 390 hydrogen bonds. So here if you see, Nana, that is option 1 is the correct answer. It is very simple. You have to do in that way, then you will get it easy. Next, transduction was discovered by Lederberg and Zinder on Salmonella typhimurium. This is discovered on Salmonella typhimurium, directly as from molecular basis. Next question, if you take again crop varieties, their hybridization, selection of diseases, it is given directly from the strategies, ma. Directly from strategies, where he asked us to find out mismatch. Again, if you can see mismatch, that is from option 1 and 3. Here these 2 areas you can find the mismatch. It is directly from the book, memory based. Here if you see identify the incorrect statement, this is from plant physiology. This is from plant physiology, wherein proton gradient across the membrane is for ATP synthesis, and that proton pump you are calling it as ATP synthase. Next, fixation of carbon dioxide in C3 plants. Fixation of carbon dioxide in C3 plants is synthesis of 3-phosphoglyceric acid, true. Next, photophosphorylation, hexose sugar synthesis, wrong. Photophosphorylation is not synthesizing hexose sugar, okay. Next, carbon dioxide fixation by pepcase, that happens in the C4 cycle, wherein it is involved in synthesis of oxaloacetic acid. So here we have to look into option 3. Next question, if we see, this is from the gene expression, okay. This is from the gene expression of molecular biology, wherein here if you see, first this is promoter. To the promoter all the time RNA polymerase is binded. To the promoter RNA polymerase will bind, and I gene will release out the repressor. I gene will give repressor, fine. To the repressor lactose will bind. To the repressor lactose will bind. And here we have 3 structural genes. Z is coding for beta galactosidase. Z is coding for beta galactosidase. And A is coding for transacetylase. A is coding for transacetylase. So if you see here clearly, A is repressor, B is inducer, that is lactose, C is beta galactosidase, and D is transacetylase, that is option 2. Now if you see, okay, clearly if you look into A cell placed in hypertonic solution, after some time its osmotic potential is measured as minus 0.5 megapascals. Then what will be the water potential of it? Always do remember, if water potential of the solution is same for the solute, when that solute is placed in the another solution, here also water potential will be same, because it will attain the equilibrium. It will attain the equilibrium. So here what will be the answer, ma? Option 2 will be the correct answer. And next if you see list 1, 2, 3, This is with respect to transgenic plants again from strategies. So if you can see from strategies, there are 3 questions given this time directly from the table. Okay, so don't skip that topic at all. Okay, now study the following list and identify the correct matching pair to Kelvin cycle. Kelvin cycle they have given. So here in the Kelvin cycle, if you see, number of ATP and NADPH required to fix 1 molecule of carbon dioxide in C3 plants. For 1 molecule of carbon dioxide, it is 3 ATP and 2 NADPH are required. Second is 1 molecule of glucose in C3 plants require 6 CO2, 18 ATP and 12 NADPH. Third is ATP and NADPH required to make 1 molecule of glucose. Only assimilatory power he is asking. So that is 18 ATP and 12 NADPH. Number of carbon dioxide molecules required to form 1 molecule of glucose are 6 CO2s are required and 6 Kelvin cycles are required. So here the option will be option 4. This is from gene mapping. Next question, this is from gene mapping, wherein if you take a chromosome, here the genes are mapped. Whenever gene mapping question is given, you have to take those 2 genes which has high recombination frequency. You have to remember recombination frequency is always directly proportional to the distance between 2 genes. So if you take A and D, okay, A and D will have 24% between C and D, and A and D is having, A and D is having 29%, which is highest distance. First you have to map those 2 genes which has long recombination frequency or long distance. Next is C and D, okay, C and D, it has 24%. And then if you have, that is A and B, which is 20%. A and B is 20%. And then B and D. B and D is of 9%. B and D is of 9% and A and C is of 5%. So whenever genes are given, you have to map from more distance genes to less distance genes. That is, if you clearly locate the genes on the chromosome, it is ACBD. ACBD. Okay. Next. Which of the following or which seeds cannot germinate and establish without the presence of mycorrhiza? Mycorrhiza is obligate symbiotic association between roots of the pinus plant and fungi. Okay, so here the answer will be pinus because it has obligate relationship with the fungi. Fine. The roots of fungi are in obligate association with, the roots of pinus are in obligate association with fungi. This is from biotechnology. Sir will be dealing to you that question. Next, assertion and reason, if you see, from seed dormancy. Seed dormancy may be caused by hard seed coats in the Fabaceae. True, seed dormancy is caused by hard seed coats in the Fabaceae. Such type of seed dormancy is broken by stratification. Wrong. Such type of seed dormancy is broken by scarification. So here assertion is true and reason is false. Next, complete maps or genetic maps was first prepared by Alfred Stuyvesant, that is option 2. Next, identify incorrect statement. This is from mineral nutrition, where we have to remember this. First is zinc. Zinc is for ABA synthesis, correct. Next, so here he has asked us to find out incorrect statements. First we will identify the correct statement. Boron for cell elongation is true. Best example, pollen tube elongation. 3. Nickel for urease activator, correct. So if you take zinc and molybdenum, those 2 are wrong. So this question again from biotechnology, sir will be dealing beta. This again from biomolecules, wherein hydrogen peroxide is catalyzed by the enzyme catalase and the prosthetic group is heme. Okay, catalase, and the prosthetic group is heme. Next, under zoology, I would like to discuss two questions which are from genetics. Okay, and after those two questions, sir will be dealing zoology for you. Just a second. Here if you see 73rd question, that is from blood grouping beta. If one parent is having A blood group and the other is having B blood group, these blood groups are not expected in their children. Which blood group is not expected in the children? Okay, so both A blood group and B blood group are homozygous. They are homozygous means both IA alleles are only there. Okay, so when they are crossed, what are the possibilities? What are the possibilities of getting only IA, IB blood group? Okay, there are the possibilities of getting IA, IB blood group. So, what will be the possibilities? The blood group which is not expected in their children is A, B, O, because all the offsprings have only one chance of getting AB blood grouping. Next. Here if you see, study of the following and identify the correct statement. The alleles IA and IB regarding the blood group are dominant. IA and IB are dominant. True. IO over IO, IA, IB. They are co-dominant. Okay. So first here if you can see, there is a comma over here. That is how we have to look into the statement based questions properly. Wherein IA and IB are dominant over IO, correct. IA and IB are co-dominant, true. So statement 1 is true. In some fishes, reptiles and birds, females are heterogametic, correct. Males are homogametic, true. Genic balance theory states Y chromosome is essential, wrong. Genic balance theory states X chromosome is essential for determination. Next, person with O blood group are called universal donors, true, because RBC contains both antigens that is A and B. So here we have to choose the correct statement, that is option 1 and 2. That is option 1 and 2. These two are correct. Okay. And next is of zoology only. So now I will call upon Ramesh sir to deal the complete zoology paper and few questions from biotechnology in botany. This session will be highly useful for all the AP, EAPCET aspirants and for all 11th and 12th class students as well. Okay, so watch it carefully, where every concept will be covered here. Sir, please. Yes, continue, sir.
Hello, my dear aspirants. I'm going to discuss about zoology. Yeah, I'm going to explain about each question. Yes. First one. Yeah, like the study of fossils of animals. Like study of fossils. Fossils are what? You can come across in the Jurassic era. There is a Jurassic like dinosaurs. Okay, the fossils. Our like scientists, they can find the fossils, and that is the study. Now, so here the study of fossils, paleontology, and yeah, they confuse you guys. Similar names: paleobotany and paleozoology and evolution. Here you have to observe the study of fossils of animals. Animals mean zoology. Okay, very easy. Okay, you have to find the animal is zoology. Like, if come to the next question. Next question. Pick up the sacred groves from the following. It is a biodiversity question. There is a question every year regarding biodiversity. This is about the Aravali Hills and Chanda. Aravali Hills and Chanda are the biodiversity hotspots. Like, wait for a moment. एक minute. Yes. Yeah. Here, look at this. Yeah, Aravali Hills and Chanda is the biodiversity hotspots, like sacred groves. Biodiversity sacred groves are the Aravali Hills and Chanda. It is the direct question from the NCERT. Yes. And next one. Yeah, this is sort of innovative question. Yeah, the bilaterally symmetrical animals are more efficient than others, seeking food, locating mate and escape from these enemies. Bilateral symmetrical animals. Like, there is a formation of head mean there is a neurons, they can together to form in the anterior end. That is the cephalization. Cephalization is going to take place. Because of cephalization, there is a sensory neurons, or they can observe the surrounds. Does it mean there are some locating mate and seeking food and escaping from the animals? This is the advance in the nature. This is the two statements are correct. Assertion is the bilateral symmetrical animals, they are more efficient than others for the seeking food and locating mate and escaping from the enemies. This is the advanced in nature because of cephalization. Because of cephalization. Cephalization is the sensory neurons where they can observe the surrounds, like eyes. Okay, this is what head. Cephaly mean head. Cephaly mean head. Okay, because of the nature is advancedness. Next question. Both A and B are correct in this statement. Next question. Sorry. Yeah. Yeah. It is vagina is lined by. Vagina is lined by. This is human reproductive system, female reproductive system. Vagina is lined by. There is no transitional epithelium. Of course, that is not. Columnar epithelium also not. Stratified with keratinized epithelium and stratified non-keratinized epithelium. Which is one is right? Which is the stratified non-keratinized epithelium. Stratified non-keratinized epithelium. This is also direct. Okay? Yes. If come to the next question. Yeah. Next question. Yeah. Consider the following statement. Bones are homeostatic reservoir of calcium and magnesium and phosphorus. Calcium, magnesium, phosphorus. Bones are the reservoir of this, okay, these minerals, and bones are highly vascularized. Of course, bones are that is connective tissue. Connective tissue nothing but vascularized. Okay, which statement is correct? Which one of the following? Consider the following statement. Bones are homeostatic reservoir of calcium, magnesium, and phosphorus. Bones are highly vascularized. What is the statement here? We can observe. Just look into this. Yeah, 1 or 2 is right. Of course, bones are connective tissue, highly vascularized. There is a mineral source of calcium, phosphorus. Yes. Obviously, this is the question which we think, and this is easy. Yeah, if come to the next question. Yeah. Study the following, choose the right combination. Combination mean here, this is the question about match the following. Yeah, nematoda. Nematihelminthes mean askhelminthes, round worms. Nematoda mean round worms. Nematihelminthes, which is the gland, rennet gland is their excretory organ. Excretory organ in the nematoda is rennet gland. The characteristic features here, this is the group, and the example, okay, ankylostoma. Ankylostoma, nothing but it is the nematoda. And if come to the crustacea, crustacea is arthropoda. Arthropoda. Arthropoda, there is a excretory organ is green gland. Arthropoda, there are types of excretory glands are there. Malpighian tubes, green gland, coaxial glands. So, if come to the crustacea division, there is a green gland is the excretory organ. This is pelamen. Is the example. Yeah. Cephalopoda. Molluscan. Cephalopoda is ink gland. Ink gland is the excretory organ. Sepia is the example. Okay. Yeah, it is animal diversity one. The direct question they did ask. Okay. No confusion. Echinodia. This is Aristotle lantern. Of course, you guys know. Yeah. Echinus is the example. So if come to this, this is the correct, this is the correct, and this is the correct. This is also correct. Match the following. All are correct. Yes. Just look into this. Fourth answer is the right. Fourth is the right answer. Next, tapeworms belongs to. Tapeworms nothing but cystoda. It is a flatworms. Okay. Tapeworms nothing but flatworms. Directly question is cystoda. There is no need explanation for this question. Yeah. If come to this, the animals are commonly called scavengers of the sea. This is the question which is you must know. Scavengers. Okay. That is hagfishes. Hagfishes are the scavengers. This is also direct question. No need explanation for this. This is the example question. Okay. Yep. And come to the next question. This is also match the following question. Match the following question. If you know, if you good at two options, that is enough to eliminate remaining. Yeah. If come to this, just look into this. Yep. The right systemic arch. Okay. If come to the human, this is the heart. The left systemic arch. Left systemic arch come to the human mammals. Temporophagy, it is obviously reptiles. Vocal sacs is obviously frogs. Frogs can be vocal sac, especially male frog. And yeah, right systemic arch is completely birds. There is no bone fishes in this option. Just look into this. A is 3, B is 4, 5, and C is 1, and D is 2. Yeah, this is the option. Yeah, second one is right. D is 2, C is 1, B is 5, A is 3. This is the right. Yes, next one. Yeah, this is the direct question. The number of flagellae in trichomonas. Number of flagellae in trichomonas, it is 8 in number, sorry 4 in number. This is also direct question. Okay, yes. Next, choose the hyperparasitic. This is also example, animal diversity 1. This is also direct question. Hyperparasite, Nosema notabalis. Nosema notabalis is the direct example question. This is no need further explanation. Yeah, just look into this Plasmodium falciparum. Yeah, Trypanosoma gambiense. No. Okay. Yes. And this is the question. Consider the following statements. The crop of female Anopheles microgametes of the Plasmodium are separated from the microgametocytes by the exflagellation. Micro is the male, microgametogenesis male exflagellation is going to take place in the male. So the crop of female Anopheles microgametes of the Plasmodium are separated from the microgametocyte by exflagellation. Yeah, of course, this is the right answer. The Schiffner's dots are believed by the antigen caused by Plasmodium. Of course, Schiffner's dot, this is the test, the malarial test is, they can conclude this is the malaria or not under the test is Schiffner's dot only. Schiffner's dot are the antigen released elements, that is like iron content like products, which is they produce from the antigen, iron content pigments, that is Schiffner's dots. So here first is correct, second is correct. 1 and 2 are the right answer. 1 and 2 are the right answer. Both are the right answer for this question. Yeah. If come to the next question, this is also match the following question. Okay, this is pre-erythrocytic cycle. Pre-erythrocytic cycle. This is the Plasmodium life cycle. Pre-erythrocytic cycle is going to take place in the liver, of course, in the hepatocytes. Cycle of Golgi, which is cycle of Golgi is take place in the human erythrocytes. Okay, human erythrocyte. Sporogamy and gametogamy. Sporogamy is the wall of the crop of the mosquito, and gametogamy the crop of the mosquito. Okay, yeah. A is 2, B is 4, and C is 5, and D is 3. Sorry, gametogamy is D is 3. This is the right answer. Second is the right answer. Second option is the right option. Okay. Yes. Next one. Yeah. This is the direct question. Heroin is obtained from. It is a type of drug which is from the morphine. This is the direct question. Yeah. These structures hold the food and bring to the mandibles in the cockroach. The food they can take into the mouth. Okay, what is that? Maxillae. They can hold and they can take the food into the mandible. Mandible means there is a mouth. Okay, this is the direct question. Maxillae. The like cockroach mouth parts, if you know, this is the question, very easy. Yeah. Next question. Respiratory system of cockroach is polynuclear type. All spiracles are functional. Just look into this. Yeah. So spiracles are the tubes which is like in the human respiratory system, like alveolus. Like this in here, there is a tubes. they can enter from the all segments, they can exit from the all segments. The air will enter into the spiracles. All are functional. The statement is correct. And this is the polypneumatic type. Of course, the like cockroach respiratory system polypneumatic type. Okay, yeah, both are correct. Yeah, next question. The ommatidium of cockroach is the refractive region. Yeah, that is obviously cornea. Refractive region is the cornea. These are the cockroach. Questions came from the cockroach. It is 4 questions coming from the cockroach, which is structural organization. Yes. Next question. Yeah. Cyclical season of the morphological variations in certain organisms called cyclomorphism. 1 minute. Yeah. This is the ecological question. Okay. Cyclical seasonal morphological variation in certain organisms called cyclomorphosis. There are different kind of region on the earth. There are polar region, temperate region, tropical region, okay, equatorial region. According to their like cyclical seasons, they can change their body structure and their lifestyle. This is the cyclomorphosis is the right answer. If come to the Allen's rule, the state that with the increase of the every 10 centigrade, the rate of metabolic activities is double. Is it Allen's rule? No, it is enzyme activity. Enzyme activity increase for every 10 centigrade double. What is Allen's rule? Allen's rule says there is certain regions, the size of the animal, okay, either increase or decrease. The appendages mean their wings or their like nails, such a skin cover. So Allen's rule says the size of their appendages will increase or decrease based on their environment. Okay, they can changes. There is a modification based on their environment in the winter season, in the like summer season. Seasonally, their morphology, like in the like species, they can change their appendages, mean their body extra things. Okay, they can change according to the environment. They can adjust according to the environment. That is Allen's rule. So statement 2 is wrong. The statement 2 is wrong. The enzyme activity will increase every 10 degrees centigrade up to the optimum temperature. That is enzyme activity. Allen's rule, they can change their body structure according to the environment. This is what in the second statement. First statement only correct, second statement is wrong. Yeah, here is the 3 is the answer. First is true but the statement 2 is false. If come to the next question. Yeah, and Ranatra and Notokia. 8 minutes. Yeah, Ranatra and Notonicta, the names in an ecosystem are desired as. Guys, nectans. Here answer is nectan. What is nectans? Nectans are highly speed movement in the water surface. Nectans. Okay, they can move highly speed. Okay, yes. And next question. Choose the greenhouse gas. It is methane. Methane is the greenhouse gas. Okay, this is a direct question. Greenhouse gases are there, methane and carbon dioxide instead of carbon monoxide. Methane and carbon dioxide are the greenhouse gases. Direct question in the environment. Next question. Next question. Aminopeptidase secretes by the pancreas. Yeah, aminopeptidase secretes by pancreas and as well intestine also. Intestine also. So this statement is wrong. And enamel on the teeth is secretes by amyloblast cells. Of course, this is right. Dental formula in the human milk dentation is 2102. Of course, there is no premolar and one reduce of molar, and this is the formula is 2102 2102. This is the milk teeth formula. There is absence of premolar. And liver is the largest endocrine gland. Just look at this endocrine they mentioned. Liver is the largest gland, but endocrine largest gland is thyroid. Thyroid. So this is the wrong statement, and first one is intestine also. So first one wrong and fourth one wrong, and 2, 3 is the right answer. They did ask wrong. They did ask the correct statement. 2, 3 is the right answer. Correct option. Yeah. Volume of air in the lungs even after forceful expiration. Yeah. Volume of air after forceful expiration mean there is a remaining after forceful expiration. Remaining volume is they can call residual volume. This is about the lungs volumes. Okay, there is a question. This is the important concept for the exam point of view. Residual volume. Yeah, next question. Amphibians and reptiles are the blood circulation is described as incomplete double circulation. Yeah, amphibians and reptiles, 2 atrias and 1 ventricle is there. This is the underdeveloped of the division between the ventricles. So if the blood enter into the ventricle, it is mixed, oxygenated and deoxygenated. There is no 2 chambers in the ventricles, only 1. So it is incomplete double circulation, right? It is a mixed circulation, mixed blood. And they have single ventricle, yeah, of course, single ventricle in the heart, where oxygenated blood, deoxygenated blood are mixed and circulation in the body parts mixed blood. Of course, both are true. Both are true. R is the right explanation for the A assertion. Okay. So first one is the answer. If come to the next. Yeah, rotifers, earthworms and terrestrial arthropods and crustaceans, and here excretory organs and their animals. Yeah, crustaceans, green gland as we discussed the previous question. Terrestrial arthropods, of course, Malpighian tubes. No, Malpighian tubes. Terrestrial arthropods is the Malpighian tubes. Earthworms, Metanephridia. Earthworms, Metanephridia. Rotifers is the Protonephridia. Okay. A3, B5, A3, B5 and C1, D2. D2. Yeah, fourth one is answer here. Just look into this. Fourth one is answer. Yeah. If come to this bone of skull is modified into incus in the middle ear. Middle ear there are 3 bones, malleus, incus and stapes. This is the incus is modified skull bone. Okay. What is that? Which is quadrate. Which is quadrate. Okay, this is the innovative question. In the skeletal system, quadrate is the right answer. Yeah, here also observe the following and find out the related part of the brain. First one, foramen oval. Foramen oval located between the heart chambers. Between the two atrias, there is a hole we can call foramen oval, which is closed after birth. We can call fossa ovalis, and that is first one is not right answer for the related brain. And second one, foramen Monroe. Yeah, this is about brain. Foramen Monroe is about brain. And ductus silicus also, ductus sylvicus, sylvius, this is also related to the brain. And ductus arterius not about brain, it is about blood vessel. Metacele also brain. So brain, it is 2, 4 and 6. The answer is 2, 4 and 6. Yeah, if come to the next question. Thymosin is secreted by thymus gland, play a significant role in the strengthening of immune system. Of course, thymus is the this is the organ which is the lymphoid organ which is involved in the immune system, thymus. They play a major role in differentiation of T lymphocytes. Absolutely right answer. They can relevant to the T lymphocytes only. Both are true, and that is the explanation. R is for A is the right explanation. Yeah, next question. These are the hormones and their source. Prolactin, progesterone, melatonin and calcitonin. Prolactin, which is produced by, prolactin which is produced by here pituitary gland. Progesterone is produced by obviously corpus luteum. Melatonin obviously pineal gland. Calcitonin is the thyroid gland. Prolactin A4, A5, B3, A5, B3. Just look into this. Yeah, A5, B3 is the fourth is the right answer for this question. Very simple. Okay, in the match the following questions. Yeah, connective tissue monocytes become histiocytes. Just look into this. Blood is the connective tissue. Okay, monocytes in that histiocytes. Okay, this is the innovative question. Okay, the connective tissue is monocytes become histiocytes. Yeah. Kupffer cells are that is about liver, microglia and memory cells. No, that not connective tissue. Yeah, Sertoli cells, Leydig cells, corpus luteum and luteinizing hormone. Just look into this. Sertoli cells. Choose the correct option. Choose the correct option is here combination. Sertoli cells, luteinizing hormone, spermiation. Sertoli cells, they can give the food for the luteinizing hormone, not a spermiation actually. Leydig cell, obviously they can produce testosterone, and this is the secondary sexual characters of male. Obviously this is right. Correct option. Corpus luteum, estrogen, no, it is progesterone. So it is wrong. Spermiogenesis, no, oogenesis. Luteinizing hormone, Graafian follicle, ovulation. This is the right. So here 2 and 4 is the right. Yeah, 2 and 4 is the right option here. Yes. Yeah, next question. Choose the mismatch pair. Yeah, gonorrhea is the Neisseria gonorrhea. Herpes, yeah, genital herpes. Here, just look into this. Genital herpes, human papilloma virus? No, it is the carcinogenic. Human HPV is carcinogenic. Cancer factor. Cancer factor. Okay? Yes. And just look into this. Human simplex virus is the genital herpes reason. Human simplex virus. HSV, not HPV, it is HSV. This is the right answer for this question. They did ask mismatch pair. Okay, HIV, human immunodeficiency, syphilis is trypanosoma. Okay, this is the second one is the right answer for this question. Next. Yeah, in hymenopteran insects, sperms are produced by mitosis. Of course, you can look into honeybees. Okay, male hymenopteran insects are haploid, obviously right. Okay, so both are correct. That is, R is right explanation for the A. This is the, you can go through the honeybee, the like production of honeybee, there is a hymenopteran insects. Okay, this is important question, innovative. Yeah. Yeah, blood group, A blood group homozygous and other B blood group also homozygous. These blood groups are not expected to in their children. Which blood group? AB or A and B? A, B and O also. A, B and O also. This is the right answer for this question. Is 3 is the right answer. And this is regarding, madam did explain it about botany question. Yeah, intermediate between the reptiles and birds. There is a interconnecting link between the reptile and birds. This is Archaeopteryx. That is like bird like, yeah, there is a like underdeveloped of the bird, we can call Archaeopteryx. Yeah. Next question, this is innovative question in the evolution chapter. Yeah, Darwinism explains the survival of the fittest but not the arrival of the fittest. Correct option, they did ask. Of course, first one is right. Mutations are subjected to the natural selections. Yeah, obviously mutations are the natural selections. If the fittest mutation, mutated gene, they can carry forward to the next generation. Yeah, Hardy-Weinberg equilibrium is applicable for the small population. Absolutely wrong. Hardy-Weinberg equilibrium says there is no evolution. No evolution. No evolution. If the small population is there, there is a chance of evolution. So there is the statement Hardy-Weinberg's equilibrium. There is a allelic frequency carry forward to the next generation. Next generation is stable. So this statement was wrong for the small population. Industrial melanism was best studied for the Drosophila melanogaster. It is absolutely wrong answer. There is a, in the book, there is a picture also for moth. Industrial melanization is moth. Yeah. So first and second right answer in this question. Yeah. It was more man-like. Ramapithecus is the similar. Yeah. Yeah. Similar for the, yeah. It is more like man-like ape. Ramapithecus is the more man-like ape. Yeah. The practice of mating of animals in the same breed but have no common ancestor on either side of the pedigree of 4 to 6 generations. So we can call it as outer crossing. Yeah, insulin polypeptide A, polypeptide B of the number of amino acids. Yeah, obviously it is direct question from the book. A is the 21 and B is the 30 amino acids. Tall wave of ECG indicate, tall wave T ECG indicate. In the ECG, this is the ECG, tall wave of T, P Q R S T. If the T is increased high, this is the like contraction of ventricle which is long and relaxation is not end in the T. Okay, that is we can call hyperkalemia. This is innovation question, important question. Yeah, guys, zoology questions are completed. Okay, yeah, the chemist sir will take care next session for the chemistry questions explanation. Thank you. Yeah, chemist sir will take care of this session. In the coming session, he will handle just 1 minute.
Okay. So, hello everyone. I think everything is clear here. Voice, video is properly visible and my audio is clearly audible. Everything is good, right? So let's start our chemistry part here. So we will be discussing chemistry part now. So let me directly go to chemistry first. So I hope you already watched last session also 2025 NEET EAPCET question papers of 29th afternoon shift we discussed. I hope you watched that video already. So if you haven't seen it till now, so after this session you can go and watch it. It will be there in our playlist. Okay, so do not skip because these sessions are very useful for your exam. Okay, So do not ignore. We will be doing especially for those students who are going to attempt for AP EAPCET. Okay, so watch it and you can get something from this content. Okay, so here we have chemistry part. Okay, so which is online paper. So here 40 questions are there, all the 40 questions should be attempted. Okay, so let's see and let's start our session. Here first question we have, according to Bohr theory of hydrogen atom, the approximate angular momentum of electron in hydrogen atom. Okay, so how can we do this kind of questions? Okay, so here we have, look here, so this is a question I am discussing here. So let me check once everything is going well or not. Yeah, so this is a question we are discussing here. So Bohr model, so according to Bohr model, angular momentum we need to calculate. So how can we do for hydrogen atom in ground state? So angular momentum L equals to, we have NH by 2 pi. So only those orbits are allowed in which their angular momentum is an integral multiple of h by 2 pi. We are studying about this now in the Bohr model. So where n represent the energy levels or the shell numbers, right? So since they are saying ground state, so n equals to 1. So angular momentum L equals to 1 into, what is the value of h here? Planck's constant, they only mentioned here, take it, 6.62 into 10 power minus 34 joule second, okay, by 2 into, what is the pi value? So pi equals to 3.14, okay. So you can cancel out this, therefore we can get it is around 0.5. So angular momentum equals to L equals to 1.05 into 10 power minus 34 joule second. So this is our answer. So do we have any option like that? Yes, 1.05 into 10 power minus 34 joule second is the second option. I hope it is clear now. So let's move further. So 122, question number 122. Okay, so observe the elements from hydrogen to calcium, and how many elements have no unpaired electrons in their ground state. So how can we do this? So how many elements have no unpaired electrons? So you can take this hydrogen atom first. Okay, so hydrogen atom, what is the electronic configuration? 1s1. They are saying how many electrons, how many elements have no unpaired electrons? No unpaired electrons. See, it has one unpaired electron. Okay, they are saying no unpaired electron. So no unpaired electron means beryl, if we take the second group elements, hydrogen, lithium, all this contains one unpaired electrons because their general electronic configuration is ns1. So like this, unpaired electron should not be there. Okay, they are saying no unpaired electron, but here we have one unpaired electron. If we say second group elements, what we have? Beryllium, magnesium, these things now. So second group elements if we take, so they have ns2, there is no unpaired electron here. So these two elements, because this is atomic number 4, this is atomic number 12. So like that we have to start from atomic number 1 to atomic number 20. Okay, same. So based on this model only, there is one question in the 29th afternoon shift. Okay, this is 29th morning shift, that is 29th afternoon shift. Both are containing same level of questions, and you know, same concept they have covered here. Okay, so it is better you have to watch complete video, complete lecture of this, and also you can try questions from that topic also, that paper also. Therefore you will feel somewhat confident. Okay, so you can also confirm, you can just confirm and tell me in the comment box whether two papers are same or not. Okay, similar or not. Okay, so from atomic number 1 to 20 if we consider like this, so second group elements have 0 unpaired electrons, next third group will be having, 13th group boron family will be having p1 electron. So like that, it means in p orbital one unpaired electron. Like that if we consider again in halogens, no unpaired electrons, like not in halogen, in inert gas we have no unpaired electrons because helium is having ns2 configuration, right, and the next neon, argon. Okay, so these two having ns2 np6 electronic configuration. Okay, right. Okay, so what are the elements here? We have helium, neon, argon, here beryllium, magnesium. Is there any other element Again, so they are saying up to calcium now, so you have to take calcium also, 20 elements. So up to 20, these are the elements which are having no unpaired electrons. So these are 3 and here are 3. So total how many? 6, that's it. So we have option number 1 as 6, that's it. That is your final answer, right? So let's move forward. Among lithium, sodium, sulfur, oxygen, lithium, sodium, sulfur and oxygen, the elements have most negative electron gain enthalpy. So we know more electronegative atoms will be having more electron gain enthalpy, more negative electron gain enthalpy. So out of sulfur and oxygen, so down the group electron gain enthalpy increases for second period and third period elements due to large size, due to large size than this oxygen. Sulfur is having more easily it can accept the electron, so therefore it releases more energy. So I can say the tendency to accept the electron is more for sulfur than the oxygen, therefore more negative electron gain enthalpy is for sulfur. So write for sulfur, then next oxygen. Now out of this lithium and sodium, these are alkali metals, right? So down the group metallic nature, what happens? Metallic nature increases, they have less tendency to accept the electron, more tendency to release the electron. Here electron gain enthalpy means what? So tendency to gain electron. So if it is having more tendency, more energy will be released, right? So but lithium is small size, more effective nuclear charge will be there, easy, like relatively it will be having more tendency to accept the electron than the sodium, therefore I am writing lithium, sodium. So choose the option which is having in this order. So what they are asking us, most negative is for sulfur, least negative is for sodium, that's it. These are the two species, okay. So you can choose the option C, option 3 is having sulfur, sodium, that's it, that is your answer, okay. Next, enthalpies, ionization enthalpies of X, Y, Z are given. They are saying that atomic numbers are successive, so one after the other, okay, and their electron gain enthalpy values are like this, okay. So like this. So now we need to identify which elements these are. So X is having relatively higher electron ionization enthalpy, then Z is having more electron gain enthalpy than the Y. So it means maybe there is a stable electronic configuration for Zx, therefore it is having high electron ionization energy, it is having high ionization energy. So now we know in the first group they see, just see, so lithium, like sodium is given first, but X is having maximum, but sodium will be having minimum in the given order, because sodium is alkali metal, easily loses electron, therefore first option is incorrect. So if we go for this, X is having highest, so noble gases are stable electronic configuration containing species, so they will be having highest ionization energy. Then minimum for Y, so that's why it is alkali metal, because alkali metals are having low ionization energy than the second group elements, alkaline earth metals, because they are having NS2 electronic configuration, this having NS1, okay. So this fully filled s orbital electron is strongly bounded to the this magnesium, nucleus of this atom, therefore it is hard to remove the electron when we compare with the alkali metals, therefore this order is correct, okay. So neon maximum, magnesium, then next sodium, that's it, this is our order, okay. So that is there in the option number 2 only, based on the values, okay. So let's go further. So here you can see what they mentioned, which of the following statements are correct. So increasing order of covalent character for these 3 substances, okay. So these are 3 are ionic compounds only, but every ionic compound there will be some covalent nature, what they have, some covalent nature. So this covalent nature is explained from Fazan's rules. What is that? Fazan's rules, okay. So what according to Fazan's rule is, cation should contain small size cation, okay, small size cation, and large size anion, okay. So this resulting composition, this resulting combination will give us more covalent character compound. So that compound will be having more covalent character. Now look here, chlorine, chlorine, chlorine. So anion size is same, but cation sizes are different. Here it is having +2, here it is +1, this is +1. Okay. Now Mg is having like more positive charge, so small size. Okay. So this is having small size, so small size cation with anion will be having more covalent nature, right? So 1 is correct. So out of rubidium and sodium, sodium is smaller in size, so the respective cation small size with 1 anion, therefore it will be having more covalent nature than this last option. Okay, the order is correct. So first option is correct. Okay, so multiple options they are asking. Okay, then second option, high boiling point of water is due to intramolecular hydrogen bonding. So this is because of intermolecular. What is that? Intermolecular, but not the intra. So you can eliminate option 2. Let's go with option 3. Water has more dipole moment among ammonia, water and HF. Yes, that is true. Okay, you can go with 1 and 3, which is option number 1. Okay, let's move further. Identify the pair in both molecules do not possess same type of hybridization. So it is easy again from chemical bonding. So what it is saying here? Water. So what is the hybridization of water there? It is sp3. For SO2 it is sp2. So how can we do? So just take the central atom. How many valence electrons are there in the central atom? So here oxygen, sulfur, both belongs to 16th group now. So valence electrons are 6. So take 6 electrons like 6 dots. 3, 3, 6 over. So how many atoms are there here? 2 hydrogen atoms. Okay, here 1 hydrogen, here 1 hydrogen. So this is one set of lone pair, this is another set of lone pair. These two are sigma bonds. So total number of sigma bonds are 2. Total number of lone pairs are again those are also 2 lone pairs. So total hybridization number will become 2 plus 2, 4. So that is sp3 hybridization. Hope it is clear. Now for SO2, sulfur having 6 electrons, write it in this format. Okay. So now this oxygen is divalent, so to write 1 sigma bond, 1 pi bond. Similarly 1 oxygen again sigma pi and 1 set of lone pair here. 1 set of lone pair. So just 6 electrons you can take 1, 2, 3, 4, 5, 6. So this is a lone pair. So totally how many sigma bonds? 2 sigma bonds plus 1 lone pair. So totally 2 plus 1 3. So sp3 means 1 is S, remaining are P's. sp2 hybridization. Like that you can do. So for PCl5 it is sp3, for ammonia it is sp3. So they are asking not possess same. So in the first option they do not possess same, they have different, but in second option they possess same hybridization. In third option also they possess sp2 hybridization. So sp2, that is also you should not consider. So BCl3 having sp2, NCl3 having sp3. So first and fourth options are correct. So you can go with the last option 1 and 4. Okay, that is our answer for this question. Let's move further. Okay. So PV graphs. PV equals to nRT you can take and you can do this. Okay, what they are asking, the two slopes, the slope of two lines. Okay, so if you solve this, like you can take just take PV equals to nRT and you can get this from it. Okay, so from these isotherms you can draw. So isotherms means what I can say, P equals to RT by V, right? So from this is a slope we can get. So from that if you solve, if it is 1:2 ratio, you will be getting 2000 as the answer. From this you can take the ratio by drawing the graph of P versus V. We are remaining with RT, RR cancel, you can get the T which is in the 1:2 ratio. From that T1 is 1000, so T2 you can calculate, which is 2 into T1. So you will be getting 2 into 1000, it will be 2000, that's it, that will be the answer. Okay, I hope it is clear, right? So let's move further. Here again, so temperature is mentioned, 100 ml of 0.05 molar copper plus 2 ion is given, added to 1 liter of 0.1 molar Ki. What is the concentration of Ki in the solution? So what in the resultant solution? So you just take Cu plus 2, to this we are adding Ki. Okay, so now what we can get from here? So from here we will be getting Cu. So if we take 2 moles and if it is taking 4 moles, we will be getting Cu2 I2 and I2 is liberated. Okay. So for every 2 moles we require 4 moles of Ki as per the equation here. Okay. Now what we have? So what concentration of so aqueous solution? See, 2 moles required 4 moles of Ki. Na? So how many moles they have given? Molarity formula what we have? N by V. So number of moles means M into V we can write. Okay. So those are given as 100 ml milliliters into 0.05. So 5 into 10 power minus 2 I can write. So this will become 5 millimole. How many millimoles of copper? It is 5 millimole. So for Ki, so it is 1 liter. It means I can say 1000 ml into 0.1 molarity. Okay, so you can cancel this. So we are having 100 millimole. Okay, so for every 2 moles we required 4 moles of Ki. For every 1 mole we required 2 moles. It means 1:2 ratio. For every 5 moles we required 10 moles of this substance. Out of 100, only 10 moles is reacting. Okay, 10 millimoles is reacting. So again, how many is unreacted? Remains unreacted. So 90 millimoles are unreacted. So that is what we need to calculate here. So we have 90 into 10 power minus 3 millimoles of Ki is present in the resulting solution. Okay, that is what the extra concentration we have. Okay. Now this concentration, this many moles present in total volume of 1 liter. It means 1000 ml, 1000 plus it is 100. So totally 1100 ml we can consider here. So how much I am taking? So 1100 if I take, 1100 ml if I take, you can multiply with 1000. So 0 0 cancel. So I can write 1.1. So this I can also write it as, so I can also write it as 0.09. Okay, 3 zeros we should insert now. 90 into this means we can write like this, and in the denominator we have 1.1. So that is what the option here we have. Okay, so whatever the format they mentioned, you try to convert into that, so you will be clear with the options. Okay, so let me check, anyone in the comment box writing anything? No one. Okay, so you can write anything. If you are having any difficulties, you can mention in the comment box. Okay, then let's move further. Identify the correct statement from the following. Energy of an isolated system is constant. Okay, is that correct? Yes, I feel it is correct only. So what the first law of thermodynamics says? See, energy of an isolated system is constant, right? And second law we will be studying that entropy of a universe or entropy of an isolated system always tends to maximum or increases. That is what the second law says, and this is what first law. It is also correct. Okay. Now, in a closed system, exchange of both energy and matter takes place. No matter exchange, only energy exchange takes place. So this statement is incorrect. So enthalpy is exothermic process. That is correct. 1 and 3 are correct. So option 2 is correct. From the data given, 1 and 3 are correct. Okay. I hope it is clear. Let's move further. So this is a reaction given. So equilibrium reaction, and it is having Kp value as 4 into 10 power 10. Okay, this is the Kp value. Now they are asking about Kp dash by giving this equation. So just observe these two equations which are taking place at same temperature. So what is the relation between them? See, it seems to be reaction is reversed. This reaction is reversed and multiplied with 3 and divided by 2. That's why here 2 is there now. So in order to remove this 2, complete equation is divided by 2, right? Right, completely divided by 2, and complete equation is multiplied by 3. Okay, after doing this, so what is the process they did? So they multiplied with 3 divided by 2. So totally it is 3 by 2. Okay, after doing this, they reversed the equation. So whenever an equilibrium reaction is reversed, the equilibrium constant will become inversed. So Kp dash will be equals to Kp dash will be, so Kp dash will be equals to, reaction is reversed I am saying now, so it means equation, this value will be inversed, you can write Kp. Okay, now complete equation is multiplied with what? Multiplied with 3 and divided by 2, right, that's it. This is the thing you have to consider. Now what is the value of Kp? So Kp is 4 into 10 power 10, overall 3 by 2, like this we can write. Okay, so now Kp dash will be, so 4 I can write 2 power 2, 1 by 2 power 2 into 10 power 10 into 3 by 2. So see, I can cancel this 2 and this 10 as 5 times, 5 2s are 10, that's it now. So now we got finally 1 by 2 into 10 power 5, we have multiplied overall with a cube. Okay, so what is a 2 cube? It is 8, so 1 by 8 into 10, 3, 5 3 is a 15, so 10 power 15, like this you can write. Now there is no option like that, so we need to change it into some other format. So 1 by 8 into 10 power 5, 15, I can write it as, so 1 by 8 into 10 power minus 15, so here I am taking 10, so I should divide with 10, so I am taking 10 power minus 16, like that. So this will becomes 1.25. Okay, so there will be an option like this, 1.25 into 10 power minus 16, that is our final answer. Okay, let's move further. I hope everything is clear without any confusion. Yes, let's move further. So for stabilizer, see, H2O2 hydrogen peroxide easily decomposes, like to form H2O and oxygen gas is liberated from it. Okay, so that's why in order to make it to not undergo decomposition, to stabilize it, we will be using stabilizers. So stabilizer here we will be using is urea. So what is a compound we will be using as a stabilizer? Urea is a stabilizer used for this H2O2 as a stabilizer. Okay, that is a direct question again. Again here also sodium bicarbonate, yes. So in fire extinguishers, sodium bicarbonate is used. Okay, so sodium bicarbonate present in the fire extinguisher, that cylinders. So at the top there will be sulfuric acid. So this, whenever that knob is pressed, that sulfuric acid will falls into the sodium bicarbonate, and the reaction between this bicarbonate and sulfuric acid takes place and liberates CO2 gas, so which is used for the stopping or to put off that fire. Okay, so that is what the purpose of this sodium bicarbonate. Yes, this statement 1 is correct. When it comes to statement 2, so like Na2CO3, so that is used as a softening, used for softening of water. See, we know hard water, soft water is there now. So in order to remove the hardness of water, so that line Na2CO3 is used. Okay, you can remember that. Okay, that is also correct. So NaOH is used in the purification of bauxite, that is also correct. So all 3 statements are correct. So we have option 4 which is having all 3 statements are correct. Okay, then next, among the alkaline metal, the metal having least melting point, that is for magnesium. Okay, you can go with option number 3, because so here melting point depends upon interactions, I mean what kind of interaction it has, metallic nature as well as the crystal lattice structure also. So due to that reason it will be having lower melting point. Okay, so diborane, see guys, so guys, I before the exam, before this TS EAPCET itself, I have made a special video on diborane. So those who watched the diborane video, which is just a 10 minutes video, I know there will be at least one question. Yes, this is the question came in the exam. Okay, so if you haven't watched, there is a series called Golden Minute series in the playlist of our YouTube channel of this NEET World. So just go and look into it. Okay, so there I have covered this very clearly, so you can watch it and you can enjoy that session, so that you will get complete clarity about diborane. So now they are asking about industrial process of preparation of diborane. So what is the industrial process? So this will be the industrial process. All these processes are correct, but this is the industrial process. This is only industrial process in the given 3 options. So you can go with option number 1, which is having 3, right? This is based on inert pair effect. So group 14 is given, 14th group elements are given. They have electronic configuration as NS2, NP2. This is the general electronic configuration. So now down the group, only NP electrons will take place in the reactions, but not NS. Only the top elements in the periodic table will participate in, will take place for all 4 electrons. But down the group, tendency to show reactivity in bond formation will be only for NP electrons, but not for NS electrons. This is what the inert pair effect. How can we define the reluctance or the inertness of NS pair electrons to take part in chemical bond formation during the chemical reactions is called inert pair effect. Okay. So due to that reason, down the group, +2 oxidation stability increases for group 14 elements and +4 oxidation state stability decreases. That's why, so if we see +2 oxidation state stability increases, I am saying down the group, and +4 oxidation state stability decreases. So down the group, these are the elements: carbon, silicon, germanium, tin, lead, these are the elements. So down the group, their +2 oxidation stability increases. This is correct. And here both are ionic in nature. PbI4, see lead, what I told you, that compounds will be like PbX2 type only, they will exist in the stable state. PbI4 does not exist because here it is having +4 oxidation state. So that is also correct. All 3 correct statements. 1, 2, 3, all 3 are correct here. Okay, next, go through the next question. So increase in concentration of X in the pregnant woman, in the blood of pregnant women who have habit of smoking may induce a premature delivery. What is X? That is carbon monoxide. Okay, that is a general question. Carbon monoxide. What is the IUPAC name of this compound? So how can we give? Here we have methyl, here we have methoxy group. So both are substituent now. So both are substituents means methoxy, as per the alphabetical order, methoxy comes first, right? So I can go, so the general name, if we do not have this, so I can say if there is methyl group, so we should call it as, if there is methoxy group, we should call it as anisole. Upon, like on benzene ring, if methoxy group is present, we should say it as anisole. If this one is there without methoxy, if only methyl group is there, we should say toluene. Okay, so now we should call it as anisole. Okay, 1, 2, 3, 4. So 4 methyl anisole. So 4 methyl anisole. We have option number 3, which is 4 methoxy anisole. Okay, 4 methyl anisole. So sorry, 4 methyl anisole, which is option number 3. And the next, here we have phenol, like hydroxy group and amine. So we know priority is more for alcohols. So we can call it as phenol ring with third position amino. So 3 amino phenol. So 3 amino phenol. That's it. Both options here. Option number 3 is correct for this particular question, right? I hope it is clear. This is the correct one. Next, guys, in the last paper also there is a question on KMNO4. There I think 2 or 3 questions are there in KMNO4. Again this question paper also there are almost 4 questions on KMNO4 oxidation reactions. Just on this one, just look into the discussion. This is one question. I will show you the remaining questions also. Check here. So butane is given. So KMNO4 they have treated. So always remember when this butane, this is alkane, na butane. So whenever we have an alkane, so like alkane I am taking, it is converted into oxidation process. I mean, so this is an oxidizing agent. So whenever we have alkane, so these tertiary, like this KMNO4 will only oxidize tertiary hydrogens only to respect to alcohols, like this. Okay, like this you can write. Okay, this alcohol treating with phosphoric acid. Okay, so treating with phosphoric acid means, so it will be undergoing dehydration. Remove one water, OH from here. Okay, so if it is undergoing dehydration, we will be getting a product. So okay, so what kind of product we are getting here? That is a question here. Okay, H3PO4. Okay, So let's see what's happening here. So this is the way we can say how many carbons are there. See, 4 carbons are there here. 1 2 3 4 5 carbons I have written, that's way. So let's retake this initial product, initial reagent. See, so first take 4 carbons. Okay, if I take like this, there is no tertiary carbon na? So what I should do now, we have to create a tertiary carbon compound. So like this I can take. So here we have tertiary hydrogen. Now treat this with KMnO4. What it will do? It will be giving us alcohol here. So now treat this with H3PO4. Upon heating gives alkene as a product. So this is a product, alkene as a product. Okay. So this one upon treating with KMnO4 in a heating condition. So what we got? So this is the alkene we got now. So this upon treating with KMnO4 under acidic medium. So just cut this into 2 equal halves. Here we know here CH2 group is there. So just put oxygen above and below. Therefore we will be getting this as one product. And what about top side? So we have carbon. To that oxygen should be inserted. The directly attached hydrogens are there now. That will be converted into OH. From this water elimination takes place. Like water elimination takes place, gives CO2 plus H2O. Therefore what are the product? We got acetone as a product. We got acetone. So from this what I told you, from this if you break it like this, put oxygen. So we got acetone as a product, as well as CO2 gas is liberated plus water is formed. Okay, these are the 3 things. So we have option here. Okay, that is our option. Okay, I just wanted to look into screen whether it is able to visible or not. Same story here, KMnO4 given with alkene. What I told you to do, same story you can apply. Break this bond, put oxygen here and here, that's it, done. Next, if to these alkene carbons any directly attached hydrogens are present, convert into OH group. So what we are getting? CH3, CH. If direct H is there means we should write OH. Double bond is there, convert into OH. This is the product. Both side groups are same, so we will get 2 moles of this. So this is our X. Acetic acid is our X here. Okay. The next, what about Y here? So alkynes with sodium in liquid ammonia. So undergoes reduction and gives trans alkenes like this. Trans alkene. Okay, this is called a bridge reduction. BIRCH, bridge reduction. Okay, so where do we have this kind of option? I think we do not have here. So this is correct, but this is cis alkene. So this is not the correct one. So it means option number 4 might be the answer. Let us look into it. Yes, acetic acid we have, trans alkene we have. That's it. So trans alkene is a product formed in the bridge reduction. Okay. So in order to get cis alkenes, we should use Lindlar's catalyst. Okay. PD BaSO4 H2. Okay. Next. So an alcohol X, there is an alcohol X, okay, X let us say. So this treated with HCl, okay, at room temperature, gets converts into corresponding chloride. Okay, so we will getting some chloride here, some alcohol chloride. So this X, okay, again this X upon dehydration means removal of water. Okay, dehydration, this is alcohol. Okay, followed by ozonolysis gives Y and Z. So gives Y and Z. So Y followed by ozonolysis. Ozonolysis gives Y plus Z I am writing. Okay, let us look into it. Okay, so now reaction is taking place at room temperature. So what is this X, Y, Z we need to find here. What are Y and Z? So we need to identify X first. So at room temperature alcohol giving alkyl product means with how many carbons? 4 carbons. So I am taking tertiary alcohol, upon treating with acid forms, like remove this water molecule first and substitute here chlorine. So this is the tertiary chloride formed. Okay, this is happens in the room temperature only. Okay, so now this upon dehydration, here removal of water they are saying now, X upon dehydration. So here methyl group hydrogen and this OH you should remove as water, therefore we can get this alkene as a product. Now this alkene on ozonolysis, whenever you have ozonolysis process, just break double bond and insert oxygen both sides. Only oxygen where we have double bond, there you have to insert. So one is acetone, another one is formaldehyde. Acetone, formaldehyde, first option is correct, right? I hope it is also clear without any confusions. Okay, so place, this is the CH now, so place one oxygen upside downside, that's it, to the double bond. So this is happens in the ozonolysis process. I hope it is clear. Let's move further. Identify the crystal system in which body center lattice is not present. So body center in tetragonal, orthorhombic, cubic, body center will be there. In hexagonal, no body center, you can see. Okay. Then next, mole fraction of sulfuric acid in its aqueous solution is 0.9. What is the mass fraction? Okay. So now mole fraction of sulfuric acid is given. So mole fraction, let us say for sulfuric acid is 0.9. So how can I write mole fraction? So mole fraction is nothing but number of moles of sulfuric acid by total number of moles, N total we can write. Okay. So this is 0.9 means 9 by 10. Okay. Now how many moles of sulfuric acid here we have? Here we have is 9 moles by comparing this. Okay. So now from this we need to find mass percentage now. So that it means we require what is the mass of sulfuric acid first. So for that 9 moles are there. So moles we can write, moles of sulfuric acid we can write weight by its molecular weight equals to 9. So what is the weight now? 9 into molecular weight of sulfuric acid, that is 98. 98 is the sulfuric acid molecular weight. Okay. We got weight of sulfuric acid. Now total weight means we have sulfuric acid as well as aqueous medium. So we know the total weight of sulfuric acid, we need to find weight of water here. So weight of water means how many moles of water is there? So if it is 9 moles of water, out of this 10 moles, 9 moles of sulfuric acid out of this 10 moles, so 9 over, it means remaining one is water. So total number of water molecules are 1 and its molecular weight is 18. So 1 into 18 I am writing directly using this formula. Okay. So total weight is how much? 18 plus 9 into 98. This is the total weight. So in the numerator we should write weight of that component, whatever we want to find its weight fraction. Okay. So in this 9 into 98, okay, into 100. So if you do this, you will be getting around 98%. This is our answer. So how can we do? This is simple formula. If you want to find a weight of sulfuric acid, a percentage weight of sulfuric acid, you just take weight of sulfuric acid by total weight of all the components, total weight into 100, that's it. This is the formula I have used here. Okay. So we will be getting option number 3 as the answer for this question. Let's move further. Okay. So let me take some water here. Okay, let me check what are the questions we have. Okay, fine. Here for a first order reaction, A gives rise to P. So in this reaction the following graphs are given. So they are saying first order now, so rate is directly proportional to concentration of A we can write. So dA by dt, this is what the rate equation, right? So now we know the slope is okay. So greater the slope, I mean greater the value, so greater the slope, correct order of slopes. Okay. obtain the slope set point A, B, C are given. What is the correct order of the slope? Yes, so for first order it is more for A because greater the slope there. So MA is greater than MB greater than MC. Okay, you can directly understand it from the formula. So what is the integral equation here we have? So from that also, that only you can explain 1 by T, like this we have now, 1 by T ln A naught by AT, concentration at time T. So from this you can answer this question. Okay, then let's move further. Sorry. Give me some time. Okay. So following Freundlich isotherm absorption isotherm was obtained for a gas. Okay. So now the experimental condition for the graph between C to D. So from C to D. So how it is looking? It is a straight line. Say straight line like see here. So it is a straight line now. So it is straight line means it reached the saturation point. So I can say saturation due to high pressure of gas. So what is this? So due to its high pressure of gas. Yes, saturation of adsorption due to high pressure of gas. So here we have option number 2 for this question. Okay, so that is why the graph is like a straight line here. Okay, so you can also focus on Longmire adsorption isotherm and Freundlich absorption isotherm. What is physisorption, what is chemisorption from adsorption chapter. Okay, surface chemistry. Okay, so you can focus on these 4 topics. What is physisorption, chemisorption, what exactly adsorbent, absorbate, and the next Freundlich adsorption isotherm and Longmire adsorption isotherm. These are the 4 things you should focus from surface chemistry. Okay, when it comes to this question, which are the following metal is not correctly matched with its refining process. So here it is, vapor phase refining is not the correct one for copper. So remaining are correct only. Okay, then next for this match the following. Ozone, SO2, sulfuric acid and phosphine is given. So what are their uses? So here phosphine will be used for smoke screen. Okay, so in the warfare, so I can say D4. Next ozone is used as germicide, A3, D4A3 we have in first option. Okay, so let us SO3, sulfuric acid used in storage, batteries. Okay, so it means SO2 used as anti-clone. So do we have any option? We need to check. So what we got? A3B1, A3B1C2, A3B1C2. Yes, we have option number 3. So you can choose that option, A3B1C2. Okay. Then next, acidic strength. What happens? Remember here. So HF down the group, fluorine. We have fluorine, chlorine, bromine, iodine. These are the elements present in the halogen, down the group. Okay, so small size fluorine having small size hydrogen, so their overlapping is very strong, therefore bond strength is more. Okay, so therefore it is bond strength is more, so it will not give hydrogen easily when compared with the iodide and hydrogen iodide. So due to the large size iodide, small size hydrogen, the overlapping is very weak, so it will easily dissociate and gives H plus ion easily. Therefore which is more acidic? HI is more acidic. Down the group acidic nature increases, strong acid is HI. This order is correct. Okay, why? The bond dissociation enthalpy of hydrogen halide is following this order. So HI require minimum energy, easy to break, easily gives H plus ion. Okay, I hope this is also clear. Both statement, assertion, reason both are correct. Okay, so let's look here. Assertion, reason both are correct. Okay, first option is correct. Next, among the following, among the noble gases, the most abundant in the atmosphere, that is what? Argon. Okay, this is a direct question, right? Next, the change in oxidation state when sulfur during the oxidation of thiosulfate in neutral alkaline solution of KMnO4. So what we can get here? let me take yellow. So SO, no, not yellow, let me take some pink. Okay, so sulfur, thiosulfate now. So thiosulfate means it is Na2S2O3. So S2O3 I am taking. Sodium thiosulfate means Na2S2O3 we can say. Okay, so now when upon treating this with KMnO4, okay, so it will be in neutral or faintly alkaline medium, they are saying neutral alkaline medium. Okay, so here it is +2 oxidation state. So this converts into sulfate. -2 means here it is oxidation state is +6. So +2 to +6 conversion taking place. So here +2 to +6 conversion taking place. First option is correct. I hope this is also clear without any confusions, right? Okay, let's move further. IUPAC name of this complex, metal complex is given. So first of all, if you consider this coordination sphere which is in the anionic cationic position, now what are the ligands we have? Just identify them first. After that, follow their alphabetical orders. This is amine, this is aqua, this is chloro. Which alphabet comes first? Amine, AM comes first. So write tetra aqua, tetra aqua, tetra amine, amine, tetra amine. Then next aqua comes now, A in aqua. So aqua, no space in between the letters, in between this sentence like words. Okay, next chloro, aqua, chloro, next chloro. Okay, next cobalt, metal you should write cobalt. Okay, cobalt. So what is its oxidation state? Right here, see 2 chlorines outside, inside 1 chlorine, so totally +3 will be the oxidation state. So 3, cobalt 3, and outside the coordination sphere we have chlorines now. So leave some space, little space, leave a space and write cobalt 3 chloride. Okay, this is the name. Okay, so where do we have tetraamine, aqua, chloro, cobalt 3 chloride? That's it. This is our answer. Okay, that's it. So intermolecular forces, increasing order of intermolecular forces. So for this, more dipole movement, I mean dipole interactions, more polar is a compound, more it will be having more interactions. So 1 is more, then 3, then 2. So fourth option is correct for this question. Okay, then next. So vitamin C and vitamin E are sources for vitamin C and vitamin E are. So for vitamin C, citrus fruits we can say, and amla also correct. But for vitamin E, sunflowers seeds or sunflower oil is the best option. So option 2 will be correct for this. Okay, you can go with option 2. Next, correct. Identify the correct set of from the following. So ofloxacin, which is a broad spectrum and bactericidal. Bactericidal means what? So it has both effects, killing bacteria and also inhibiting the growth of that bacteria. Okay, so that is what we can say bactericidal. But bacteriostatic means it will just it will inhibit the growth of bacteria but not killing it, right? So bactericidal. So ofloxacin having correct explanation here. So both are this is correct. Okay, so you can go with the option number 2. Okay, then next, again KMnO4 question here, again third question on KMnO4. Isopentane is given. Okay, isopentane is given. Okay, 1, 2, 3, 4 isopentane. So here we have tertiary hydrogen. Upon treating with KMnO4 gives tertiary alcohol like this. So this is our X. So this X upon treating with Y giving chlorine. So this is just use HCl concentrated like that. So we will remove that tertiary alcohol using this acid and replace it with chlorine. That's it. Substitution reaction follows SN1 mechanism. Okay, so where do we have this is the alcohol. Okay, because we know only tertiary hydrogens will be oxidized by KMnO4 from alkanes, right? So correct, concentrated HCl, this is also correct. So option number 3 is correct for this. Let's move. boiling point of diethyl ether is less than 1 butanol. Yes, so boiling point of 1 butanol having, see, always remember alcohols having more boiling point than the other things. Why they have more boiling point than these ethers and aldehydes, ketones of similar molecular weight? Because of more extent of hydrogen bonding, greater hydrogen bonding in alcohols, therefore having more boiling point than the other. Okay, so this statement is correct. And diethyl ether is miscible with water to almost same extent as 1 butanol. Actually, yes, almost it is correct only, but we have seen it is 2 butanol, no issues. Maybe diethyl ether and also 1 butanol having almost similar miscibility, so both statements are correct here. Okay, last 3 questions. Isopentyl alcohol on reaction with reagent X. So what is isopentyl alcohol? So first of all you need to know what exactly isopentane. This is isopentane. Okay, isopentyl alcohol reaction with X gives corresponding carboxylic acid. Okay, so in order to give corresponding carboxylic acid, definitely it should be a primary alcohol, so I am placing it here. Okay, now so just what we need to do, so we need to use, okay, tertiary alcohol. So you need to okay, you need to treat with some oxidizing agent, so that will give the corresponding alcohol. So let me take this and treat with KMNO4 in acidic medium. Okay, so this gives us COOH. This is a carboxylic acid formed. 1, 2, 3, 4, 5. Okay, this is a carboxylic acid formed. Okay, now from this carboxylic acid, upon decarboxylation means you have to treat this with NaOH plus CaO. Okay, upon heating, just remove the CO2 part and you will be having, so 1, 2, 3, fourth carbon is removed, this carbon is removed, 1, 2, 3, and you will be having like this. So this is one CH3 group, another CH3, another CH3, like this we got. Okay, now this is what gave Y. So this is our Y. Okay, it is also looking like Y only. Okay, now reagent with reagent Z gives tertiary alcohol. So what is the reagent? What is the reagent? See, here we have one tertiary hydrogen now. So if I use KMNO4 here in direct KMNO4, not in acidic medium, directly KMNO4 I am using. So what it will be giving? It will convert tertiary hydrogen to tertiary carbon containing hydrogen will be converts into alcohol. That's it. So option 2 is correct for this question. I hope it is clear. Here acidic KMNO4, here it is simple KMNO4 is used. Oxidation, see, KMNO4 oxidizing reagent is one of the important. So in EAPCET I felt that they are more focusing on reagent parts. Okay, there are in the same paper there are 4 questions on reagent chemistry, especially on KMNO4. What are all the application given for KMNO4 in your 12th and 11th standard books? Okay, you just need to learn those. Okay, so this is about the one thing. Okay, so if you want to know more about this KMNO4, just go through hydrocarbon chapter where I have discussed everything. I mean most of the examples which are useful for this oxidation purpose, like KMNO4 and the next ozonolysis, ozone process, and also some other reagents I have discussed there. You can watch it if you want. Okay, so that might be helpful because we have seen this is the 4th question in the same paper about KMNO4, right? And last few questions are there. So X and Y are respectively in the following reactions are what? Okay, so in order to convert into bromination of this benzoic acid, we need to use, see, benzoic acid like carboxylic acid is electron withdrawing group, so electrophile goes to meta position, that we know. So iron, like something like Lewis acid, here iron we can use metal. So and the next bromine, Br2, so bromine, iron for first one, and here methoxy group is electron releasing group that will direct the electrophile to this para position. So we can use here what, bromine with some iron metal we can use, or something like acidic medium we can use. So this is the most appropriate one. bromine with acetic acid. So bromination takes place at para position. So fourth option is correct for this. Okay. So the reagent and organic compound required to convert ethyl bromide, let us say, to 2-butanone. So this is a 2-butanone. So how do we convert here? What are the reagents we can use? Okay, so ethyl bromide is there. So upon treating this with, see, magnesium is there, no? So let us treat with magnesium. Okay, so we will be getting Grignard reagent ethyl magnesium bromide. Okay, so now this ethyl magnesium bromide, ethyl is having negative charge and this will be having positive charge. So now our ethyl is strong nucleophile. Okay, so now this nucleophile can react with this and convert directly this and this to alcohol. But what we required? Ketone is required, right? So it means only one time nucleophilic attack should takes place, not the twice. If it is taking twice, we will be getting alcohol, but we don't require alcohol, we require ketone. So it means the nucleophilic nature should be reduced. So when we treat this with cadmium chloride, we will be getting cadmium, diethyl cadmium. So we will be getting ethyl 2 times diethyl cadmium. So this is a reagent. Now using this reagent, if I take this CH3CO, this CH3COCl, CH3COCl. So now what happens is ethyl anion will comes and attacks on this which is a nucleophile, attacks on this electrophilic center, chlorine goes out. See, we are getting methyl, so ethyl is coming, this is what the ethyl. So we got the final product. So which is the best option? Option number 3, one of the very interesting question, like conceptual question. Okay. Next, this is also very interesting question and very important. You will be seeing this in all your competitive exams, whatever the exam you are writing. So there will be this point, like carbylamine test, Hinsberg test. These are the organic reagents, practical organic chemistry reactions where you will be doing in the labs. Okay. So an amine X unheating with chloroform in ethanolic forms a foul smelling. So carbylamine test is given only by primary amines, product formed is isocyanate, and it will be having very foul smell, bad odor here. Okay. So let me take a primary amine to understand more clearly. So primary amine means R, okay, on nitrogen only one alkyl group should present, okay, remaining 2 should be hydrogens. Okay. So upon treating this with chloroform in basic medium, it will gives RNC. So this is isocyanate product. Okay. So this is what the carbylamine test, this is what the product form. It will be having foul smell. Okay. So now only primary amines will react. Okay. So this is primary, this is also primary, that is fine, and they both can react. So let's continue our discussion. Y, the product of Y of another amine Y, this is our X, Y means another one they are saying. Okay. And benzene sulfonyl chloride. So this is what amines with benzene sulfonyl chloride is Hinsberg test. Okay. So this is given by primary and secondary amines. Okay. So primary amines with benzene sulfonyl chlorides forms a product, and that product upon treating with basic medium like alkaline KMO, like KOH, like alkali medium, so that will be soluble. Okay. Which one? Primary amine will, the product formed from primary amine and benzene sulfonyl chloride will dissolve in the, which will soluble in the alkali medium, whereas secondary amine will react with the benzene sulfonyl chloride and form some product, but this product will not soluble in the alkali medium. Okay. So from this statement you can read Y and benzene sulfonyl chloride reaction takes place, is insoluble in the product of the other amine Y and benzene sulfonyl chloride will be insoluble in the alkali medium. It means this is secondary amine. So one is primary amine, other one is secondary amine. See, this is primary only, this is also primary, this is not the correct one. But in the second option, one is primary, other one is secondary. So this will be the most correct appropriate answer. Okay. So this is done. Yes, we are done with the question paper of EAPCET 29th shift. Okay, 29th morning shift and evening shift both are done. So in the next session we will be discussing 30th shift, I mean the April 30th shift. Okay. So guys, I hope you really liked the session. I may feel that these sessions are very useful for your preparation. So if it is really useful, so just click on the like button and also share with your friends, those who are really preparing or those who are going to prepare for next academic year. That means your juniors, your friends, anyone. Okay. So just share with them. So thank you for watching. Happy learning. Thank you.